Hello, this is Radio Hannah, and you are tuning into the topic of digestion and absorption, which I can assure you is one of the nastiest topics on the agenda in my opinion, and therefore the sooner we understand it, the sooner we can stop panicking about it, and trust me, I would know. Today we're looking at all the different enzymes involved in our digestion, as well as its products, and then how they are absorbed. Sound daunting? Fear not, although this is a longer video than normal, just try to sit back and get acquainted. I have already done a lot of the summarising hard work for you. Let's get into it. We'll start with some key words. Again, a few more than normal, and they're very repetitive, but we're going to separate them all into neat little boxes later on, so don't you worry. First up, we have a bunch of enzymes. Amylase, which helps break down starch, then lipase, which helps break down lipids, peptidases, including exopeptidase, endopeptidase, and dipeptidase, which helps break down proteins. Next, we have a few sugars, including monosaccharides, disaccharides, and polysaccharides, which are obviously sugars and therefore energy sources we're trying to break down during digestion. Mono meaning one, di meaning two, and poly in this context meaning three or more. Next up is maltose, fructose, lactose, glucose, sucrose, and galactose, which are all either subtypes of monosaccharides or disaccharides and are just the specific names for each type. Hydrolysis is the process of breaking down bonds by adding water. Catalyzing is the process of speeding up a reaction, and emulsification is the process of breaking down lipid droplets into smaller droplets that are easier to digest. Finally, we have glycosidic bonds, which are found in starch and are broken down to produce sugars. Gee whiz. Collect yourself for a moment after all that, and we can move on. Okay, let's start with the digestion of carbohydrates. Carbohydrates are found in starchy foods like bread and potatoes and are broken down primarily by amylase. Starch is made up of polysaccharides, sugars, and amylase works by separating up these polysaccharides by hydrolysis to make maltose, a disaccharide, basically just smaller sugars. Membrane-bound disaccharides then work to reduce these sugars even further into monosaccharides, which will be easier to absorb into the bloodstream. Amylase, our first enzyme, is produced in saliva and by the pancreas, which releases it into the small intestine. Next up is lipid digestion. Lipids are basically fats found in, you guessed it, fatty foods like red meats or dairy products. The enzyme that helps digest these lipids is helpfully called lipase, and it breaks them down into monoglycerides, glycerol, and fatty acids through hydrolysis. Bile salts also help to emulsify lipids into small droplets that have a greater surface area and are easier to break down. From there, the monoglycerides and fatty acids stick with the bile salts to form micelles, which constantly break up and reform to release the products of lipid digestion to be absorbed. Lipases are primarily made in the pancreas and then secreted into the small intestine, just like amylase, and bile salts are produced by the liver. Finally, we have protein digestion. Proteins are found in food products like chicken, eggs, or nuts, and are broken down by a combination of different peptidases into amino acids. Endopeptidases like trypsin hydrolyze the bonds within protein. Exopeptidases hydrolyze the bonds at the end of protein molecules, removing single amino acids, and dipeptidases work specifically on dipeptides to split them into two amino acids by hydrolysis. Amino acids are absorbed by active transport into epithelial cell membranes. Peptidases are produced by the pancreas, stomach, and small intestine. Great. Thank you so much for listening. It's a topic where there's a lot of very similar words that you have to learn by heart, which makes it difficult to learn in my opinion, but we got through it together. Good luck in the exam, and I'll see you when I see you.